Welcome to the Foundation Frontiers educational series on comprehensive genomic profiling. My name is Jamie King, and I'm a senior medical writer at Foundation Medicine, and I'm joined by Julia Quintanilla, a senior translational scientist at Foundation Medicine. In this video, we will learn about detecting genomic alterations in DNA repair pathways with CGP and Foundation Medicine's pan-tumor HRD biomarker, HRD signature. These videos are for educational purposes only. And so, Julia, as we introduce homologous recombination, can you explain how DNA repair pathways work and how they're altered in cancer cells?
Yeah, sure. So homologous recombination is a signaling pathway used to repair breaks in DNA strands. And in cells with HR repair deficiency, also known as HRD, these breaks cannot be repaired.
And what proteins are involved in repairing breaks through this pathway?
Yeah, so there are several proteins involved in this pathway. The most famous examples are BRCA1 and BRCA2, which are tumor suppressor proteins coded by the BRCA1, BRCA2 genes. PARP proteins are another class of proteins involved in DNA repair through the HR pathway. And in cancer cells with HRD, targeting PARP proteins in cells with mutated BRCA1 and BRCA2 can lead to cell death through a process known as synthetic lethality.
Thank you so much, Julia, for the overview of this pathway. Why is detection of genomic alterations in this pathway important for treatment decisions?
Yeah, so genomic alterations in HRR genes may provide some clues to determine if a patient's tumor is sensitive to treatment with targeted therapies, like, for example, PARP inhibitors.
And so, when we think in clinical context, let's take a closer look at a hypothetical patient case to see how CGP may be applied to detect changes in HRR genes specifically.
Yeah, so in this example patient case, a 57-year-old female patient presents with advanced ovarian cancer.
And so, Julia, what types of therapies are currently indicated to treat advanced ovarian cancer?
So PARP inhibitors like olaparib, for example, are indicated as maintenance therapy for advanced ovarian cancer with somatic or germline BRCA1 and BRCA2 alterations after initial platinum-based chemotherapy.
And so, this patient was initially treated with surgery and chemotherapy, and her germline testing for BRCA1 and 2 alterations was negative. So, a tissue biopsy was available for further molecular testing. So, how might CGP be used in this situation?
Yeah, so CGP with tissue sample may detect some somatic alterations in HRR genes like BRCA1 and 2 that are associated with response to PARP inhibitors. However, not all genomic alterations in HRR genes lead to HRD, and HRD may be also caused by non-genomic mechanisms. And this patient tissue CGP test did not reveal any actionable genomic alteration. So next, we'll look at additional biomarkers reported on the Foundation Medicine Tissue CGP test that may be informative for treatment decisions related to HRD.
And in addition to the changes in HRR genes, can you tell us a little bit more about other genomic changes observed in tumors that are associated with HRD?
Yeah, so HRD can arise through various mechanisms. And tumors with HRD show DNA changes known as genomic scars, and these scars include large scale copy number changes in genes. Foundation Medicine's tissue CGP test reports a unique biomarker called HRD signature or HRD-Sig that measures scarring associated with HRD. And this biomarker was built from over 100 copy number features and expands copy number analysis beyond previous methods to allow for a pan-tumor utilization.
And what does the landscape of HRD-Sig look like across tumor types?
The HRD-Sig positivity rates in association with HRR alterations varies across tumor types. So a higher prevalence, over 30%, has been observed in ovarian cancer, while moderate rates between 6 to 17% have been observed in breast, prostate, non-small cell lung cancer, and also pancreatic cancer.
Great, thank you. And so now that we've covered HRD-Sig in general, let's take a closer look at HRD-Sig in the context of ovarian cancer and revisit the patient case that we covered previously.
Yeah, so in Foundation Medicine database, we observed variable HRD-seq positivity rates within the gynecological cancer types. And with advanced ovarian cancer samples specifically, we observed that HRD-seq status could be assessed for 24% more samples than another measure of HRD, the GLOH.
And what about HRD6 correlation with clinical outcomes in ovarian cancer?
In analysis of real-world data, we observed an association between HRD-seq positive status and improved real-world progression-free survival with maintenance therapy with a PARP inhibitor in patients, even among those patients with wild-type BRCA.
And so, how might these observations be informative in the example patient case where no actionable HRR genomic alterations were found from the tissue CGP testing?
So, in this ovarian cancer case, where no actionable alterations were observed, but HRD-seq status was positive, this may be informative for the clinician to consider PARP inhibitor therapy, since HRD-seq positive status is correlated with clinical benefit, even in the absence of BRCA or other HRR alterations.
And great. So, when we think from a broader perspective, how is HRD-SIG informative for HRD in cancer?
So, HRD-seq is a scar-based biomarker that can provide more information beyond simply looking for genomic alterations in HRR genes. So, since it is a functional readout of HRD, this may help inform decisions for patients where genomic alterations in HRR genes are not present, or conversely, where HRR alterations are not directly causing HRD.
Thank you so much, Julia, for joining our educational session today to discuss the DNA repair pathways and the HRD-SIG biomarker. And thank you, everyone, for joining. We look forward to the next Foundation Frontiers educational series.
Thank you for having me.